Good morning. In this video, we are going to see how the carbonic acid bicarbonate buffer system helps buffering of the pH of the blood and helps resisting significant changes in the pH of the blood. And as we said before, this buffer system consists of a weak acid, which is carbonic acid, and we have previously said the definition of a weak acid. It is an acid that dissociates weakly in a solution, in an aqueous solution, and meanwhile, it is an acid that releases a small number of protons in that solution. But it does not only consist of the weak acid, it also consists of the salt. So the salt of carbonic acid would be a bicarbonate salt, for example, and it is the salt of carbonic acid. It should be the salt of carbonic acid from a strong base like sodium hydroxide. So actually it is a carbonic acid bicarbonate buffer system, but actually it is carbonic acid sodium bicarbonate mainly buffer system inside the human body. Why is this carbonic acid bicarbonate buffer system important? Because it is the most abundant and the most effective inside the human body. Why is it the most abundant in the human body? Because it mainly depends on carbon dioxide, meaning that in order for carbonic acid to be formed, and therefore in order for bicarbonate to be formed, then we should start by carbon dioxide. And needless to say that carbon dioxide is a gas that is very abundant in the human body, simply because it is the product of cellular respiration. Whether this cellular respiration is aerobic cellular respiration occurring in the presence of oxygen, or anaerobic cellular respiration occurring in the absence of oxygen. Both of them produce carbon dioxide. If you have carbon dioxide inside the cell or inside blood, and of course in an aqueous environment where water is abundant, then carbon dioxide and water can combine together, can react together, carbon dioxide as a gas and water as a liquid. So it is a gas dissolved in a liquid to give this weak acid, which is called carbonic acid. As we said before, carbonic acid is a weak acid. It weakly dissociates into protons and bicarbonates. And because it weakly dissociates, this means that it is incompletely dissociated, and that we are going to use this double arrow between carbonic acid and the resultant or the products, which are the protons and the bicarbonate ions. This double arrow or two-sided arrow indicates that the reaction is reversible, meaning that it occurs both ways. This means that carbonic acid can dissociate into protons and bicarbonate ions, and by the way, the bicarbonate ions will carry a negative charge, and similarly, the protons and the bicarbonate ions can reunite together to form carbonic acid. So we have a forward reaction where carbonic acid dissociates, and we have a backward reaction where carbonic acid is reformed once again. At the beginning, if we begin with carbonic acid, then it will dissociate into protons and bicarbonate at a high rate, because we have a large amount of the reactant, which is here carbonic acid or the reagent. You needn't have another substance for this to be a reactant, and it will give the reagent molecules are still abundant, so they give a big number of product molecules. But after a while, that differs, a period of time that differs from one equation to another, we will find that the products will reunite to form the reagent. And at a certain period of time, we will reach a state of equilibrium. What is this state of equilibrium? The state of equilibrium is when the rate of the forward reaction is equal to the rate of the backward reaction. This reaction is called a reversible reaction. So what is a reversible reaction? A reversible reaction is a reaction that can occur both ways. It can occur in the forward direction, and meanwhile, it can occur in the backward direction. At the beginning, the forward direction has a high rate, and the backward direction has a slow rate. And then gradually, as the reagent molecules diminish and the product molecules increase, the rate of the forward reaction diminishes gradually, and the rate of the backward reaction increases gradually until they become equal to each other, and we reach a state of equilibrium. It is worth mentioning that this equilibrium is called dynamic equilibrium and not static equilibrium. Why is it called dynamic equilibrium? Because actually the chemical reactions do not stop. Neither the forward reaction stops nor the backward reaction stops. All what happens is that they end up by having the same rate. So apparently, things are stable. The concentration of carbonic acid stays almost the same. The concentration of protons in the medium of the reaction stays the same, and the concentration of bicarbonate stays more or less the same. But this doesn't mean that the two processes, the forward and the backward ones, are not proceeding or are not going on. After having studied this part of the chemical reaction, we return again to this reaction. We usually say that enzymes speed up chemical reaction. Enzymes are biocatalysts. They are biological molecules present in our body cells that speed up chemical reactions. But upon talking, or when we talked about this chemical reaction of dissociation and reformation of carbonic acid, we did not talk about any enzyme. So although most chemical reactions in the human body require enzymes so that they can occur at a considerably rapid speed that is good for the human body or that is suitable for the human body to achieve its functions on time, yet this dissociation reaction and reformation reaction of carbonic acid occurs spontaneously, meaning that it is independent of enzymes. It does not require an enzyme to start, neither to proceed. On the other hand, if we come back to this chemical reaction between carbon dioxide and water, we will find out that this chemical reaction is again a reversible reaction with a forward reaction, which is the one where carbon dioxide and water form carbonic acid, and a backward reaction where carbonic acid dissociates again into carbon dioxide and water. But this time, the biochemical reaction necessitates the presence of an enzyme. That enzyme is called carbonic anhydrase enzyme, and that enzyme is present in the blood and in many body cells, but in particular in red blood corpuscles, which are the red blood cells, but we call them corpuscles because they are spherical and they are non-nucleated. And it's also abundant in lung cells. And because the carbonic anhydrase enzyme is abundant in these two cells, then this buffering system will work mainly inside red blood corpuscles, inside the lung, as well as inside the blood in general. Okay? In order to understand how this buffer system works, we should first understand one important property of the reversible reactions. Let's look at the bottom of the slide. If we have a reagent A and a reagent B, for example, that reversibly unite to form substance C, and if substance C reversibly dissociates into D and E, so substance D is considered, I'm trying to simulate the reaction above, I'm trying to simulate those reactions. So these are the products of the reaction. So if you remove any of these products from the medium of the reaction, then you are favoring the forward reaction because C will try to dissociate more to make up for the absence of D and E. Similarly, on the other hand, if products D and E increase, then it is the backward reaction that will be favored, because if D and E are now abundant, then they will do everything to react and give back product C. And this is the basis of how the bicarbonate buffers or the carbonic acid bicarbonate buffer system works. We said before that the pH of blood is mainly dependent on the hydrogen ion concentration. So if it happens, for example, that you have a lot of hydrogen ions that are added to your blood, so if you add protons to your blood, for example, when you eat a lot of dairy products like milk, yogurt, cheese, or if you eat a lot of meat, poultry, and fish, proteins in milk and proteins in meat will give you acidic organic acids that will release hydrogen ions. So whenever the hydrogen ions are increased in the solution, there is a tendency for the pH of your blood to diminish. And if the pH of your blood diminishes, then the pH of the surrounding extracellular fluid and therefore the pH of the cytosol will be affected because they are interrelated, as we said before, and this would impair or affect negatively the function of the cells, because it would affect the optimum pH of the enzymes, or the enzymes wouldn't be at their optimum pH. So if you eat a lot of acidic, of a lot of foodstuff that are converted into acids and hydrogen ions are added to your blood, then this will favor the backward reaction in which protons and bicarbonates will react with each other to give you carbonic acid. You would tell me, so what's the difference? I had a lot of hydrogen ions in my blood, and these hydrogen ions combined with bicarbonate to give me another acid. So I didn't work up the issue. No, you have worked up the issue because the new acid you obtain, which is the carbonic acid, is importantly is a weak acid, and therefore it will dissociate very weakly and give you much fewer protons than above. So it's true that you haven't removed all the surplus hydrogen ions, but you removed as much as you could from these hydrogen ions. In such a case, we said that the bicarbonate ion, which united with the protons, with the surplus protons, we call this bicarbonate ion, we call it a proton absorber because it helped you get rid of the hydrogen. On the other hand, if you eat a lot of vegetables and fruit, then you will have a lot of alkalis, or for example, hydroxyl radicals in your blood, or as we said before, for example, the citric acid that is present in the citric fruits, and although it is an acid, yet it will be converted into, in the blood, it will react with sodium to give rise to sodium citrate. And sodium citrate will elevate the pH of the blood. So if you eat a lot of citrus fruits, although they are acidic, although they are sour, but in the body they give you a lot of sodium citrate, which is alkaline and which raises the pH. So now we have more alkalinity or less acidity. It is as if the hydrogen ions were removed from the solution. So this will favor the forward reaction and favor dissociation of carbonic acid into protons and bicarbonate. So in such a case, carbonic acid will be a proton giver or a proton donor. So in cases of acidity, bicarbonate acts as a proton acceptor, whereas in cases of increased alkalinity of the blood and a rise in pH, we have a proton donor. What is the proton donor? The proton donor is carbonic acid. What is the proton acceptor? The proton acceptor is the bicarbonate ion, the bicarbonate ion. So you can see that the proton donor is a neutral molecule, whereas the proton acceptor is a bicarbonate ion that is negatively charged. And now we are going to discuss extra information about buffer systems, but you needn't memorize this. You needn't study it for the test. It's just to make you understand more why a buffer system is formed of a weak acid with a strong base, or the reverse. Now, in case of the carbonic acid bicarbonate system, we have a weak acid, which is carbonic acid, and its salt from a strong base, which is the strong base was essentially sodium hydroxide. And when sodium hydroxide combined with carbonic acid, it gave rise to sodium bicarbonate, which is the salt of a weak acid, carbonic acid from a strong base, which is sodium hydroxide. Imagine now that we added directly an acid, a strong acid to the blood, like sulfuric acid. It's something hypothetical. So sulfuric acid, which is a strong acid, will react with sodium bicarbonate, and they will exchange their radicals. So the sulfate radical will be attracted to the sodium ion because it is negatively charged, whereas the bicarbonate radical will be attracted to the protons. The bicarbonate is negative and the protons are positive. So this will result in what? It will result in sodium sulfate and again carbonic acid. Sodium sulfate comes from a strong base, which is sodium hydroxide, and a strong acid, which is sulfuric acid. That's why, because the two radicals are equally strong, coming from equally strong alkalis and acids, then this is neutral. The salt is neutral. It is a neutral salt. And this is the rule in formation of a salt. When an acid reacts with a base, the rule is that we obtain a salt that is neutral because, and that's why we call the reaction neutralization reaction. But if the acid and the alkali are not of equal strengths, then the salt will be either weakly acidic or weakly alkaline. And we also obtain carbonic acid, which is a weak acid. Have we benefited? Have we gained anything from this buffer system? Yes, we started by adding a strong acid, which was capable of lowering the pH to a great extent, and we ended up by having something neutral and something weakly acidic. So this is again because I removed the strong acid and replaced it by a weak acid. This is one example that shows you why we have to use sodium bicarbonate, which comes from a strong acid from a strong base and a weak acid. On the other hand, if we add sodium hydroxide, which is a strong base, what happens? The reaction will occur with carbonic acid to form sodium bicarbonate and water. OH with H will form water, and sodium with bicarbonate will form sodium bicarbonate, of course, provided that the balance of the equation is respected. So in such a case, we get a neutral molecule, which is water, and sodium bicarbonate. Sodium bicarbonate is considered to be weakly alkaline. Why is it weakly alkaline? Because it comes from a weak acid and a strong base. So the base is stronger, so it's not neutral, but tending to be alkaline. So I started by adding a strong base, which is sodium hydroxide, and I ended up by having a neutral thing, which is water, and a weakly alkaline molecule, which is sodium bicarbonate. So we've benefited from the buffer system because we converted the strong alkali, which was capable of significantly changing the pH of the blood and raising it, to a weakly alkaline substance. And that's why we said before, we didn't say that buffer systems prevent changes in pH, but we just said that buffer systems resist changes in pH. Now we are going to talk about a property which is isomerism. What are isomers? Isomers are, first of all, organic compounds. They have the same molecular form or the same molecular formula, but they differ in the molecular structure. For example, molecular formula means, what does the molecular formula shows me? The molecular formula is a formula that shows me the types of atoms that share in the formation of the molecule. For example, if I'm talking about a glucose molecule, then it will tell me that the glucose molecule consists of carbon, hydrogen, and oxygen. But not only does the molecular formula show me the types of atoms that entered in the formation of that compound, of course, because we have different elements, but also the number of atoms of each element. So it would tell me that one glucose molecule, which is already a compound, not an element, consists of 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. So the chemical formula or the molecular formula of glucose is C6H12O6. But the molecular structure shows you the arrangement. For example, you would know that if you want to draw the molecular structure of glucose, then you would say that it looks like this: C double bond O H, and then you have 6 carbon atoms like this. Okay, 1, 2, 3, 4, 5, and then 6, and then attached to the sixth carbon atom, you have 2 hydrogen atoms like this, H2OH, and then you would have here H and OH. Okay. So this is an example of a molecular structure of a substance that consists of 6 carbon atoms like glucose. If you change, if you exchange the positions of H and OH, glucose might be converted into galactose. So actually, galactose and fructose have the same molecular formula as glucose. So they all have this molecular formula. Although they are 3 different types of monosaccharides or simple sugars that have different properties, yet they have the same molecular formula. So the molecular formula does not really give you full information about the properties of the substance, because it does not show you how the atoms are arranged in space. It just gives you the kind and number of atoms, but the location of atoms is given by the molecular structure. And why do I have to know, or why is it interesting to know the relative positions of the atoms here of carbon, hydrogen, and oxygen? Because it tells you the types of bonds that exist between the atoms. So for example, you would know that here you have a C double bond O, there is a double bond O, there is a double bond between carbon and oxygen, and you would know that the glucose molecule contains that group CH double bond O, which we call an aldehydic group, and that is why glucose is considered an aldose. Whereas on the other hand, fructose, which has the same molecular formula but does not have the same structural formula, has a structural formula that looks like this, CH2OH, and then you have here you have a C double bond O. So and then you continue the molecule. So here it does not have an aldehydic group at the beginning of the molecule, but it has only this C double bond O in the middle of the molecule, or attached to carbon atom number 2, you have a double bond, and this C double bond O group is a ketonic group, and not an aldehydic group. That's why fructose is not considered an aldose, but it is considered a ketose, meaning that it contains a ketonic group. So the molecules in different isomers that have the same molecular formula and different molecular structures, they will have different bonds, they will have different bond angles and different bond lengths. And it is important to know that bond angles and bond lengths are very important because inside every organic molecule, the bond angles, meaning like, for example, in water, for example, when we had oxygen and then we had hydrogen and another hydrogen, the molecule is kinked like this. There is a particular angle between the negatively charged oxygen and the positively charged hydrogen. This angle between the bonds, this angle or bond angle is, this bond angle is important because it is, how is it determined? It is determined in such a way to make the molecule more stable, to make the molecule more stable. And when the molecule is more stable, it is the least energy possible. And in AP biology, they love to hear this. The readers of your answers in the formal test love to hear this because living systems in general, whether they are living cells or body systems or the human body as a whole, acts in such a way, whether structurally or functionally, to spare energy, to be at the minimum energy possible. So bond angles and bond lengths will be determined in such a way that the whole molecule becomes more stable and becomes at the least energy bond, the least energy possible. So whenever bonds are directional, whenever we say that a certain bond is directional, as you can read here, this means that the atoms in the molecules assume or adopt a specific position to maximize the bond strength, but at the same time to minimize bond energy, to minimize energy. Okay? The covalent bonds are bonds that are made by sharing of electron pairs. And the covalent bonds, as we said before, are fairly rigid bonds. So when there is a covalent bond between two carbon atoms, when there is a covalent bond between two carbon atoms, this means that if you have A on that side and B on the other side, then it would be difficult for the two particles to exchange their places. So this is the definition of isomers. And now here you have the link at the top of the slide that tells you more about the isomers, and it is the link of a video from Khan Academy. And now we continue talking about isomers. Isomers, although they have the same chemical formula, like for example, let's take the example of glucose, fructose and galactose, which are all monosaccharides, glucose, fructose, and galactose. They're all monosaccharides and they have the same chemical formula. It's understandable that these molecules are isomers because they have the same chemical formula, but they have a different molecular structure or different arrangement in space. So we are expecting these molecules maybe to have different physical properties, like, for example, the melting point, evaporation. Physical properties is like, for example, a melting point, evaporation point, all these that do not involve, all these properties that do not involve chemical reactions between the substance and another reagent are called physical properties, or boiling point in a solution, for example. But the chemical properties are expected to be the same, since we have the same molecular structure, the same number, the same type and number of atoms. However, they do have different chemical properties because of three main reasons. So if you're ever asked, how come that isomers have the same molecular formula yet different chemical reactions or properties? First of all, it all depends upon the arrangement of the atoms inside the molecule, because the arrangement of the atoms inside the molecules will determine the type of bond. Is it a single bond or a covalent bond? Will determine the bond angle, will also determine the bond strength. Furthermore, the different arrangement will result in different functional groups. Functional groups. What is a functional group? A functional group is a group of atoms that reacts consistently the same way in all chemical reactions. So first of all, they have different chemical properties because they have different bonds between the atoms. And when they have different bonds between the atoms, this results in the formation of different functional groups. We have just seen the aldehydic group in case of glucose and the ketonic group in case of fructose. These are groups of, for example, the CHO group or the aldehydic group is a group of atoms that is considered a functional group, because whenever you find this aldehydic group in a compound, then this compound is an aldehyde, and therefore, or an aldose if it is a sugar, and it will react in a specific way. And every time you find C double bond O, then it is a ketone or a ketose in case of a sugar, and it will react the same in all chemical reactions. So they might have the same chemical formula, but because of the different arrangement of atoms, they have different bonds, different, therefore, functional groups. They might have similar bonds, but they might have different relative positions of atoms or groups around the double bond. And we will see this on the next slide. For example, if you have C double bond C, and then you have here a methyl group, CH3, and then here you have the rest of the molecule, which we will not specify. Whereas on the other hand, you have the methyl group here, and then you have the same group of atoms here. Let's call this anonymous 1x and that anonymous 1x. So here, the two carbon atoms are related to a methyl group, but the methyl group here is above the level of the bond, and the methyl group here is below the level of the bond. This is one compound. Similarly, you might have another compound looking like this, but having the two methyl groups on the same side, and the rest of the molecule is also X and X. So if you compare this compound number 1 with that compound number 2, you can easily deduce that their chemical formula would be the same. They will have the same type of atoms, carbons, for example, carbon, hydrogen, and oxygen. For example, we don't know what X is, but they will have similar types of atoms. They will have the same number of atoms in every molecule. But in the first one above, the two methyl groups, one of them is above and the other is below. So this is called the transversion of the molecule, whereas here the two methyl groups are on both sides, and we call this the cis version. So this is called cis-trans isomers, and number 1 will be the trans isomer of number 2, and molecule number 2 will be the cis isomer of number 1. So it's only about a certain functional group, like the methyl group, that is present on 2 different sides of the double bond. And because we have said that the double bond is rigid, there is no way that molecule number 1 changes the positions of the methyl groups. There is no way that in molecule number 1 methyl group, the methyl group above rotates around the double bond and comes below, because the double bonds are rigid. They fix the components in place. This type of isomerism is another type. So number 1 is the one we have just explained. Different bonds between the atoms results in a different arrangement in space simply, and it is called structural isomerism. But in number 2, we have geometric isomerism, or it is a kind of 3D isomerism, or stereo isomerism in space. But there is another type of stereoisomerism that is more complicated, which is called the optical isomerism. And both geometric and optical isomerisms are like stereo isomerisms, resulting in different arrangement in space. And we will see on the next slide the difference between the two and why it is called optical isomerism, and what are the medical applications of this optical isomerism. So again, here we have the isomers, and the isomers are divided into structural isomers or constitutional isomers and stereo isomers. And the stereoisomers are divided into geometric and optical isomers. Here you can easily see that propanol and methyl ethyl ether have the same number of carbon atoms. They both have 3 carbon atoms. So the first component in the molecular formula is C3. And then how many hydrogen atoms do they have? They have 8 hydrogen, they have 8 hydrogen atoms. So actually we have 3 carbon atoms, and we have 8 carbon atoms. So up to now we can build up the formula C3, and then we have H8, and then we only have 1 oxygen atom. We don't usually write the 1. The same applies to methyl ethyl ether. The only difference between the two is the arrangement in space, the structural formula. This different arrangement resulted in different bonding, and the different bonding resulted in different functional groups between the two. So again, in structural isomerism, we have, let's draw our flow chart. So we have different arrangement of the atoms, and this different arrangement of the atoms resulted in different bondings, and this different bonding resulted in different functional groups. Is it important to mention that there are different functional groups? Yes, because we have just said that the functional groups behave the same way in chemical reactions. So it is up to the functional groups that the chemical properties of the whole molecule is determined. Now, on the other hand, in stereoisomers, you can see here cis-2-butene and trans-2-butene. They are isomers because they have the same molecular formula. You can see here that CH3, or the methyl group, is present on the same side of the double bond. That's why it is the cis isomer. We can say that it is the cis isomer, whereas in trans-2-butene, it's just that the methyl group is the two methyl groups are not on the same side. They are on variable or different sides of the double bond. That's why it is called a trans isomer. Okay. So these are the geometric isomers. But when we come to the optical isomers, we will find that the optical isomers are mirror images of each other, but they are not superimposable. They are not superimposable. What does this mean? That they are not superimposable, meaning that if you want to take that molecule without rotating the molecule, if you want to take that molecule without rotating it and try to put it on the other molecule, they will not be superimposed. It's just like when you take, for example, the left side or the left part of your glove and try to superimpose it on the right side of your glove. So if you translate or if you like slide this right side of the glove and try to put it on the other side, then you will have the thumb, which is here, the thumb, the T stands for the thumb. The thumb will be superimposed here on the little finger, whereas the little finger will be exposed on the thumb. When do we have such optical isomers? Of course, why don't these isomers, optical isomers, rotate? What prevents them from rotation? Here, for example, we don't have a double bond. So you might ask yourselves, why don't they rotate? Why don't they rotate so that they can superimpose? They can't rotate because when we talk about optical isomerism, these substances are usually in crystals. They are usually in a crystalline form, and they why are they called optical isomers? They are called optical isomers because if we pass a polarized light, which means in order to simplify things, we would say very concentrated light, like laser, for example, we call it plain polarized light, where we have electromagnetic waves that are very focused, that are of the same length, and that are traveling in one plane. These optical isomers will show one of two behaviors. Either they cause dextrorotation, dextro means to the right, or they might cause rotation to the left. And that's why, for example, a substance like dopamine that is used to treat the tremors of the famous disease called Parkinson's disease, that's now very famous because it's spreading. Parkinson's disease is a degenerative disease of the brain that sometimes precedes Alzheimer's disease, and it is characterized by very coarse tremors of the hands. And these coarse visible tremors of the hands are seen when the hands are still, when they do not move, and when the patient is tense in particular. And you have the impression that if you give, for example, a cup of tea to that person, the cup of tea would drop. But these tremors fortunately disappear when we hold something, when the hand is in, when the muscles of the hand are working. But in order for treat Parkinsonism is due to deficiency of dopamine in the brain, in a particular area of the brain that is called the basal ganglia, a group of nerve nuclei or cell bodies of neurons that is full of dopamine, or that should contain a certain amount of dopamine in order to prevent this. So you treat the patient by giving dopamine. And dopa is the precursor of dopamine, and it has been found that L-DOPA is effective, the L version of DOPA is effective in giving dopamine in the basal ganglia of the brain and treating Parkinsonism, whereas D-DOPA, for example, which is the other optical isomer, is ineffective. So it is important to study, especially in medicine. I'm talking about medicine, so they are optical isomers. It is important to know that L-DOPA and D-DOPA are optical isomers and that they are not equally effective in the treatment of Parkinsonism, one of them being very effective, the other being completely ineffective. Here we can see examples of structural isomers, and you can easily deduce if you count the number of carbon atoms, the number of hydrogen atoms, that they have the same molecular formula. Let's count the carbon atoms. The carbon atoms are 4 in all of them. Let's count the number of hydrogen atoms. They are 10. So we have, we have C4H10. This is the chemical, yes, formula, the molecular chemical formula of all the 3 molecules, C4H10. But as you see, they have different arrangements. The different arrangements have resulted in the fact that this molecule, for example, which is called neopentane, has 4 methyl groups. The molecule has been arranged like this, C, with 4 bonds. You know that carbon is tetravalent. And then here, everyone had 3 hydrogen atoms like this. If we expand the structural formula and so on, I don't have space here, but see here, this is by changing the chemical, by changing the arrangement of the atoms, you have created a new functional group, which is the methyl group with all the properties of the methyl group. And it is important to study this because we will see, for example, how addition of a methyl group, for example, to your genes and to your DNA may greatly affect the activity of DNA. Whereas in pentane, for example, we don't have, actually we have 5 carbon atoms, I'm sorry. We have 5 carbon atoms and 10 hydrogen atoms. So pentane, penta means 5. We have 5 carbon atoms and 10 hydrogen atoms. Here, isopentane is just an isomer of pentane, but we don't have a methyl group. Neopentane is another isomer of pentane. These substances are called pentane, for example, is considered a hydrocarbon. A hydrocarbon is a molecule that consists only of is an organic molecule consisting only of hydrogen and carbon. So pentane is a hydrocarbon and it consists only of hydrogen and carbon. So this is a good example of structural isomerism. In geometric isomerism, we have this example. Of course, you don't have to memorize the names, but you just have to know if we give you this compound and that compound, that you count the number of carbon atoms, you count the number of hydrogen atoms, you find that they are similar, but it's clear that they have different arrangement. Take care that there is a double bond, and it is easily observable that we have 2 methyl groups on opposite here. They are on opposite sides, so it's the trans form, and here they are on the same side, so they have, it is the cis form. So they have the same molecular formula, they have the same type and number of atoms, but they have the same type of bonds between the atoms. Here there is no difference in the bonds between them, but it's only about the spatial arrangement of the atoms between them. So they are the first type of stereoisomers, which are the geometric isomers. Here, for example, in optical isomerism, if we look at that diagram, we can see that there is, here we have an unpolarized light that consists of magnetic waves in different in different planes, So here we have different planes, but here this polarizer or this screen is made of a material that helps polarization of light. So they all have the same plane. So unpolarized light means light that consists of electromagnetic waves that travel in different planes, whereas polarized light consists of electromagnetic waves that like travel in the same plane. And polarized light, what's the importance of polarized light? The importance of polarized light is that it helps you study the optical isomers. Optical isomers have the same molecular formula. They have the same type and number of atoms, of course, because they have the same molecular formula. So since they have the same molecular formula, then they have the same type and number of atoms. They also have the same type of bonds between the atoms, but they have different spatial arrangements. You might be asking yourself, and so what? Geometrical isomers have different spatial arrangements. Yes, but geometric isomers had different spatial arrangements around a double bond. But here, they have different spatial arrangements with a very important condition that their mirror images are non-superimposable. Their mirror images are non-superimposable. And this is very important. If we come back to those two optical isomers, they have mirror images, but these mirror images are non-superimposable. These mirror images are non-superimposable. Is it important that we say this? Yes, because if it happens that the mirror images of the two molecules are non-superimposable, this indicates you that the molecule is non-symmetric, And because the molecule is non-symmetric, then they are superimposable. So here, for example, if I consider this unknown part of the molecule, or let it be a hydrogen atom, as it is in the other molecule, let this be another hydrogen atom. So here you have a central carbon atom, which we call a chiral carbon atom. What is a chiral carbon atom? A chiral carbon atom is a central carbon atom, and that central carbon atom is bonded to 4 different atoms or groups of atoms, bonded to 4 different atoms or groups of atoms. So here you can see that this is a chiral carbon atom because it is bonded to bromine, it is bonded to hydrogen, it is bonded to fluorine, and it is bonded to chlorine. So this is a chiral carbon atom, a central carbon atom that is bonded to 4 different atoms. And what's the importance of having a chiral carbon atom in an organic molecule? A chiral carbon atom in an organic molecule results in an asymmetric molecule. And if we have an asymmetric molecule, then the mirror images are non-superimposable. The mirror images are non-superimposable. We said before that they cannot change their position if they are crystalline. But what happens if they are not crystalline? What would be the importance of saying that they are non-superimposable if they are not strongly bonded to each other? The importance of saying that the mirror images are non-superimposable is that this is an indication of optical difference or difference in optical activity. Difference in optical activity concerning plane polarized light. So again, if you have a chiral carbon atom, this with all the definition of the chiral carbon atom, central, bonded to 4 different atoms or groups of atoms, then you will have an asymmetric molecule. And if you have an asymmetric molecule, then the mirror images are non-superimposable. What's the importance of the fact that the mirror images are non-superimposable? This is just an indication that there is a difference in optical activity of concerning plane polarized light. One of them deviates the plane polarized light to one direction and the other one to the other direction, and this indicates a difference in the ability to treat Parkinsonism, for example. So they're identical in chemical properties. They have different optical activity. There is a left-handed version and a right-handed version. Actually, I'm sorry, it's not rotation to the left and rotation to the right, but it is anti-clockwise and clockwise. These optical isomers also have another name, which is enantiomers. So we can see here the enantiomers or the optical isomers. You can see, for example, that sometimes we have, this is also applicable in amino acids. So here we have a central carbon atom. And it is a chiral carbon atom because it is related to, or it is bonded to hydrogen, to an amino group, to a carboxylic group, and to an R radical. These are 2 mirror images and they are non-superimposable. So it's very important in pharmaceutical industry because the 2 optical isomers might not have the same chemical properties. For example, as we said before, L-DOPA is used in the treatment of Parkinsonism, whereas D-DOPA, which is its optical isomer or its enantiomer, is almost biologically inactive. And for an unknown reason, all the amino acids in the body are left-handed. And in evolution, it has been discovered that after death, amino acids will be rearranged to give the D version. And just like we did in radioactivity, the ratio of D amino acids to L amino acids in a dead body can be an indication of the age of the dead body because it takes time. Last thing to be done on that video is to compare between the isotopes and the isomers. And for this, we are going to open a new white screen to see the difference between isotopes and isomers because you frequently mix them up. So isotopes, mainly when we mention isotopes, we are talking about elements. Whereas when we mention isomers, we are mainly talking about compounds, not elements. Isotopes are different versions of the same element that have the same atomic number, but they have different mass numbers. So they differ only in what? They differ only in the number of neutrons. Whereas isomers are compounds that have the same molecular formula, but they have a different structural formula or different arrangement of atoms. In isotopes, they usually they are chemically identical. They are chemically identical because they have the same number of electrons. They have the same electronic configuration. Whereas isomers are chemically different. They behave differently, chemically different. Why? Because this different arrangement results in different types of bonds, and the different types of bonds will result in a different behavior. And then you give an example. For example, here we can say carbon 612, which is the non-radioactive, and carbon 614, which is the radioactive form. And then here you can give examples of structural, geometrical, and optical isomers or enantiomers. Thank you so much, and see you in the next video to complete our discussion on biochemistry. And in the next video, we will be starting our detailed study of organic molecules. Thank you so much.